Well, thank you everyone for joining us today. This is part of a monthly series by the School of Medicine, Basic Sciences, called Lab to Table, where we bring together scientists to talk about public topics of interest. Today, we are talking about the science of sight and advances in eye research. My name is Aaron Conley. I am the Director of External Affairs, Partnerships, and Communications for the Basic Sciences, and I will be moderating the talk today. And so just for a little bit of background, as baby boomers generation grows older, eye diseases like cataracts and age-related macular degeneration and glaucoma are becoming much more prevalent. According to the National Eye Institute, more than half of all Americans 80 years or older either have cataracts or have had surgery to treat cataracts, while 10% of adults age 50 or older have early stages of AMD, and 3 million Americans have glaucoma. So this is an important issue, and June is actually Cataract Awareness Month. So eye researchers at Vanderbilt and elsewhere are studying how the eyes form these diseases as they age, how to treat and regenerate eye tissue, and really looking towards a future for better prevention and care of these diseases. Today, we'll be talking about what are the current treatments for these diseases, how are scientific discoveries changing the future of these diseases, and how are new technologies moving discoveries forward. A little bit of housekeeping. Please place your questions in the Q&A. We received a large amount of questions in the registration process. We'll be trying to get to as many of those questions in the end of this event. And I am pleased today that we have experts in eye research from Vanderbilt and Vanderbilt University Medical Center, and I'm excited for them to introduce themselves. Dr. Rex, would you please start us off?
Well, good morning. I'm Dr. Tonya Rex. I'm a professor in the Ophthalmology and Visual Sciences Department at Vanderbilt. And my research is these days focused on glaucoma and ocular trauma and visual dysfunction associated with traumatic brain injury. But I will also address questions dealing with retinal gene therapy because I have training in that and background in that area as well.
Thank you, Dr. Rex. Dr. Furman.
Hi, my name is Sabine Furman. I'm an associate professor in the Department of Ophthalmology and Visual Sciences at Vanderbilt Eye Institute. I'm originally trained as a developmental biologist, and I received my PhD in studying eye development. So I'm not a medical doctor and I don't see patients. And in my lab, we study development of the eye and the different tissues that comprise the eye using mouse as a model system. We are very interested in how factors or signaling pathways regulate development of one critical tissue in the back of the eye, the retinal pigment epithelium, and we can also abbreviate it as RPE. This tissue is heavily affected in dry AMD, age-related macular degeneration. Thus, we have recently started to investigate how we can use the knowledge obtained from our developmental studies to promote repair processes within the RPE itself when it's damaged. This is a situation that could be related to dry AMD. Our long-term goal, or to treat dry AMD, our long-term goal is to identify pathways and potentially drugs that can be used for treatment of dry AMD.
Thank you so much. And last but not least, Dr. Shire.
Hi, everyone. Welcome. Thanks for joining us. My name is Kevin Shai, and I'm a professor of biochemistry here at Vanderbilt, but I have secondary appointments in chemistry and ophthalmology and visual sciences. Most of my career I've spent studying cataract, the opacity that happens to the lens of your eye. And more recently, we've gotten into age-related macular degeneration research. And I think one of the most interesting parts of my work is the discoveries that we've made in the lens of the eye, looking at how proteins in the lens age. Those discoveries can be applied to other long-lived tissues in the body. And more specifically, I'm talking about the brain. So it's very exciting to us that vision research that we're doing and things we're learning by studying the lens, for example, inform us on what's happening in other parts of the body as we age.
Thank you, Dr. Shai. And to really get started, I think we could help frame this discussion by talking about what are the major eye diseases that you all study and how do they affect vision? Dr. Shai, do you want to start by talking a little bit more about cataracts?
Sure. So cataracts, as I said, are when the lens becomes cloudy or opaque. It certainly affects our ability to see clearly. It is the leading cause of blindness worldwide. Fortunately, there is a treatment for this, and that is commonly performed in the United States, and that is surgical removal of the opaque lens and insertion of a clear plastic or some sort of biomaterial like lens that allows you then to see very clearly. It's one of the most commonly performed surgeries in the United States. It's very safe, but it's not totally without complications. And we can talk a little bit more about that as we get into it.
Thank you so much. Dr. Furman, do you want to talk more about AMD, please?
So in age-related macular degeneration, there are two forms. There's dry and wet AMD. Wet AMD is characterized by the formation of excessive blood vessels that leak into the eye and impair vision. And treatment options are offered by ophthalmologists to basically try to inhibit this excessive blood vessel growth. And a more advanced version of AMD and some of these wet forms also progress into the so-called dry AMD. And this leads eventually to loss of cells in the retinal pigment epithelium and also underlying cell death in the retina. And it starts out usually in the macula, in the point of best vision in the back of the eye, because that's very, it's an area that is metabolically very active and also very susceptible to damage. So there are risk factors involved with this. One really important pathological characteristic is formation of drusen, and that also further leads to detachment of the tissues and vision impairness and blindness eventually. And there's not really a treatment that resolves this issue, but there is a new treatment that seems to help prevent further progression of the disease. This is not for every patient maybe applicable, but there's a possibility that this can help. And so this is actually being done. This treatment has started at the Eye Institute here at Vanderbilt.
That's very exciting. Thank you so much for that background on AMD. And Dr. Rex, you study ocular trauma and some other diseases. Could you get into that for us and let us know what that's about?
Right. So one of the blinding conditions that I study is glaucoma, which, like cataracts and AMD, is also age-related. So this is a condition often associated with elevated pressure in the eye. We call that intraocular pressure, or IOP, and it can lead to permanent vision loss. Now, in age-related macular degeneration, it affects the center of your vision. The macula is where light is focused as it enters into your eye. So as you're looking at me in the screen, I would potentially appear blurry. You won't be able to see my face very well if you had AMD. Glaucoma, on the other hand, is a segmental and peripheral degeneration first. So it's easy to not notice it coming along. It's slowly progressive and age-related. So unfortunately, a lot of times we don't see patients until they have pretty already highly progressed disease. So we do encourage individuals who have a family history of glaucoma to please come in and be seen, just get checked out so we can detect it early and treat. Now with glaucoma, this is this elevated pressure, which doesn't always happen, by the way. There are some individuals who have normal tension glaucoma, we call it. Interestingly, even the majority of those patients do respond very well to intraocular pressure lowering drugs. And that's our first line of treatment. So those are eye drops, and it helps to lower the pressure in the eye, and that slows the progression of disease, and people can retain sight longer. When those drops fail, there are also surgical approaches that our physicians take, and those can be very successful in patients. Unfortunately, all of these approaches only address the pressure, and none of them completely stop progression of disease. They slow the progression, and not every patient responds well. And the drops, this was something, moving into this field was really eye-opening. Was that it's not a single drop, it's multiple drops of different types and with different timing, and in an elderly population this can become complicated, and I don't know a single person who likes putting things in their eyes, so compliance is a big challenge. So we need therapies that are independent of pressure in the eye and really take alternative approaches, and I can talk more about that through our discussion. And then, yes, you mentioned trauma. So glaucoma affects really the optic nerve, which is the highway between the eye and the brain. And it allows visual signals that are collected in that thin tissue in the back of the eye, the retina, that collects light, that converts it to an electrical signal, sends that to the brain. So the optic nerve is really critical. If that's disrupted, then no matter how well the eye is working, that visual information won't reach the brain appropriately, and we won't understand what we're seeing. So it's the optic nerve that's affected in glaucoma. And it's also what is affected oftentimes in trauma. So I study ocular trauma, which can include anterior segment damage, but my primary focus is in the posterior segment of the eye. So that retina, the neural retina, which is it's part of the brain, it's central nervous system, and also the optic nerve. And also with traumatic brain injuries, there's a close association with visual dysfunction, which can be due to damage to the retina, the optic nerve, or visual pathways in the brain. And unlike these other diseases, trauma tends to happen oftentimes in the young individuals, mostly young men. But there's also a cohort of elderly, who have incurred a fall and can have damage. But primarily my work is closely associated with the military and exploring how we can protect, prevent, restore vision in individuals who have undergone a trauma.
And I know we were talking before the event just about 4th of July, and you had said something very interesting about ocular trauma on 4th of July. Would you like to talk about that?
Yeah, unfortunately, as fun as it is to go see fireworks, people like to do them themselves rather than going to professional shows. And unfortunately, that can result in traumas that can be really devastating. So sparklers actually are the most dangerous. They're seen as being benign, and so they're put in the hands of children. And unfortunately, children have lost eyes and lost vision permanently from sparklers. So they are not as safe as they seem to be, and I strongly discourage their use. And then fireworks. They sometimes go off early, or maybe they're not going off and someone goes to inspect and they get too close, and it's like a mini explosion, and shrapnel can go into the eye. You've got burning of the eyes, right? Because you're looking at it, trying to figure out what's going on. And so unfortunately, our emergency rooms are quite busy the night of 4th of July from ocular traumas. So it's a very dangerous thing. Please enjoy your 4th of July. Go to a professional show, please.
No, it's safety is always a good thing on 4th of July. You've all brought up some of the risk factors in these eye diseases. And Dr. Shai, could you talk a little bit more about cataracts risk factors and how it develops? And are there genetic components? Is it lifestyle components? What plays into that?
Yeah, so it's sort of all of the above. So yes, there are genetic factors that can lead to congenital cataracts. It's pretty serious because clear vision in very young, very young children is important for eye development. And so if a child is born with congenital cataracts, we're talking about surgery at a very young age. And great care is taken to try to preserve the development of the eye as the child develops. But most of the cataracts that we talk about are age related. So the biggest risk factor is age. And so by the time we're 75, 80 years old, I think, Aaron, you mentioned it in the intro, 50% of people will have some form of cataract. The major risk factors for this would be UV exposure, sunlight exposure. So wear your sunglasses outside. Lifestyle, so smoking, diabetes, for example, is another risk factor for cataract. Obesity and poor diet, so diets that are high in fats. So I think those are the major risk factors. And so just trying to keep glucose levels under control in diabetes, smoking cessation. I don't think I mentioned hypertension, but controlling blood pressure, all these sort of lifestyle things, and most importantly, diet, rich in antioxidants, for example. All those things can help delay or prevent cataract.
Thank you so much, Dr. Shai. Now, Dr. Furman, Dr. Rex, is this the same in AMD and glaucoma? Is this really similar, or are there different factors at play? Dr. Furman?
Yeah, and it sounds very, I mean, it is similar, like, I mean, in age-related macular degeneration, age is the biggest risk factor. I mean, it's very strong. I think I read a number that it's up to 4-fold for each decade of your lifetime. Another really important risk factor is smoking. I mean, it's a very strong, it can double your risk for AMD. Diet is important. So you can actually supplement your diet with vitamins and special oils like fish oil or dark green leafy vegetables, antioxidant containing foods that can help slow down the progression of the disease. Physical activity is also important. If you can manage maybe to exercise or do some moderate activity during the week, like walking in the evening for half an hour, that also seems to be helpful. And then there is some kind of, it's not very clarified or consistent whether any metabolical burden has a strong risk association. So this is right now seems to be a little bit unclear, but it could be that this is also accelerating progression of the disease. And there are genetic risk factors. So the inheritability of AMD, there's a strong association, and one of the genetic risk factors is, for example, over variation in a locus that mediates overactivation of the complement system, the immune system, basically. So yeah, these are the most important risk factors, and maybe some areas where there's some intervention possible.
Yeah, there's also overlap with glaucoma, not UV light per se, which affects both of AMD and cataracts, but in terms, the age is a risk, antioxidants may be helpful. There's clearly a role for oxidative stress, and oxidative stress can lead to inflammation and vice versa. So, a low inflammatory, high antioxidant diet and exercise are most likely going to be helpful. There are also genetic roles. There's some clearly, genetic forms of glaucoma that are genetically linked in a very specific way, similar to some forms of cataracts. But the vast majority, again, like cataracts, don't have a clear genetic component, but there are risk factors. So yes, there's some overlap and commonalities in terms of the risk factors amongst these diseases. Obviously, trauma is a different thing. Just don't incur a trauma.
Don't incur an accidental trauma.
Yeah,
That's
right.
how do you get around that? But no, but thank you for defining all those risk factors. And it's really exciting time in science with new imaging technologies, new computational modeling structures, AI, machine learning. Dr. Shai, could you talk about how these new technological advances have impacted and revolutionized eye research, especially around your expertise in mass spectrometry and what that is and where that impacts research?
Sure, I would say that the imaging in terms of cataract diagnosis is not that affected by new technologies, new imaging. Basically, cataract is pretty easy for an ophthalmologist to detect. And however, in AMD, it's played a big role, as playing an increasing role as we try to diagnose AMD at earlier and earlier stages. Because the earlier we can find these disease processes happening, the earlier we can intervene and delay progression. So you mentioned mass spectrometry, which is what my background and training is in. So this is a technique that it's an analytical technique that allows us to determine the quantity and the structures of molecules in our tissues, in our cells. And so the place where most people in the audience might be able to think about where mass spectrometry is used is in drug testing. So performance enhancing drugs, for example, in athletes are detected using mass spectrometry techniques. So in the field of vision research, we've been applying mass spectrometry to understand, first of all, how proteins are aging in the lens and what might be causing the lens to become cloudy or opaque. And as I said in my intro, these age-related processes that we've discovered that are happening in the lens are also happening in the brain, which is just fascinating to me. The other area where mass spectrometry can play a really big role is in looking for markers of disease. So either taking a blood sample or taking maybe a tear sample. If we can detect changes with disease, this might allow us again to identify the disease process at an earlier stage. And then the last thing that I'll mention is here at Vanderbilt, the director of our mass spectrometry center developed a technique to image molecules in tissues. So we can go into a tissue, say the retina, and image exactly where proteins or lipids or metabolites are localized. And why is this important? This is important because we can then see how those molecules change with age or change with disease. And we can pinpoint the exact position of where these changes are happening. So for AMD, this is becoming really a really important discovery tool where we can find out what changes are happening in the macula. We can determine the composition of the drusen deposits that Sabina mentioned earlier and other pathological processes, and help us to understand what the biochemical mechanisms, what's happening as we age or as the disease takes foot. And then we can think about therapeutic interventions.
And I've heard imaging mass spectrometry just talked about as a true molecular microscope. And could you just give a brief sense of what's the difference between one microscope image versus imaging mass spectrometry, the amount of data and information you get?
Yeah, so for a standard microscope, you would look at your tissue and you would see outlines of cells and at really high spatial resolution, really powerful microscopes, you can get structures of different parts of the cell. But typically those types of approaches don't give you individual molecules or molecular information, which inform us on the biochemistry that's happening. So there are techniques that allow you to look at sort of targeted, I want to see this molecule only in the tissue, and that can be done. But the beauty of imaging mass spectrometry or molecular microscope, as you said, is that we can image thousands of different molecules in a single experiment, and again, pinpoint where those molecules are. And one other area of application is that the pharmaceutical industry has taken up the technology, because if they want to know where the drugs are, the therapeutics are that they're applying to the eye, either by eye drop. So if you put an eye drop on your cornea, on the surface of the eye, the question is, where does the drug go? Does it make it all the way to the back in the retina, or does it make it to the lens? And so the pharmaceutical industry has really taken on the technology to help in their delivery of new therapeutics.
Thank you so much, Dr. Rex. I think you had something to add.
Yeah, thank you. I just wanted to comment that these approaches that Dr. Shai is talking about are also very, very relevant in the field of trauma. So, while a severe or moderate traumatic brain injury can be diagnosed through MRI or CT scan and other techniques, mild traumatic brain injury is actually vastly more common. 80% of individuals with TBI have a mild traumatic brain injury. And as a field, we have a difficult time right now differentiating, a concussion, is a concussion different from a mild traumatic brain injury? And these self-reported visual symptoms that many of the individuals have, we're having a difficult time diagnosing those. So these approaches, the mass spec, and actually you touched on AI and machine learning approaches being applied to MRI is an area that I'm investigating with other colleagues at Vanderbilt and in our Vanderbilt University Institute of Imaging Science to be able to diagnose mild traumatic brain injury and those visual dysfunctions. So these areas that you mentioned are really critical to many areas in medicine and science. And I just wanted to mention trauma as another one. Thanks.
Thank you so much, Dr. Furman.
I want to add on that I think that the diagnostic imaging technologies are actively being improved right now, so that patients actually, I think they can use, eventually they will be able to use their iPhone, their phones to image their eye, and that this can be transferred to the Eye Institute for diagnosis, and that will help doing a really tight time course maybe of the progression of the disease, and hopefully also diagnose earlier, which is important in diseases like glaucoma or AMD. And I also want to bring up research-wise that there are really important technologies that have been revolutionizing the field in the past several years in terms of figuring out what kind of really molecular profile each cell in the body has. And one technique, it's called RNA sequencing, is very important to really characterize, do a very deep characterization of the state of the cell or each tissue that in disease states also is happening or occurring. And this will help us to identify biomarkers that can also maybe hopefully enable an earlier diagnosis of these diseases. So this is very important to really identify novel markers that have been not found so far, discovered so far.
And that really, go ahead, Kevin.
Yeah, I just wanted to follow up with that. So we have a program here funded by the National Institutes of Health to generate a 3-dimensional molecular map of various tissues in the body. So this is a huge consortium across the country. I think there's something like 60 different institutions involved. But here at Vanderbilt, we have what's called tissue mapping centers. So we're trying to generate three-dimensional molecular maps of the pancreas, the kidney, and the eye. And so we're trying to map where the molecules are in the different layers of the retina, in the different cells of the retina and the lens and the cornea. And again, as Sabina mentioned, it's so important to understand where the molecules are and when they're changing so we can intervene at an earlier stage of disease.
And this really leads into the next question, and we can start with Dr. Rex. But what are the frontiers of discovery for research right now? It sounds like a big part of this is gaining more systems biology, more holistic and broad understanding of mechanisms and how that translates. Dr. Rex, could you start just by describing what is basic research and what is translational research, and what are the frontiers that you're excited about?
Right. So typically when we say we're doing basic research, is that mechanistic research. And it's really, as we've been discussing, it's important to understand what those molecular changes are, really understand that mechanism, because frankly, that's where rational therapies come from. You know, once we identify what the changes are, we can begin then to understand, are they a response to the disease? Are they inducers of the degeneration? You know, what is their role of these changes? And that helps us to figure out where we need to target and how, so that we can develop therapies that are going to be effective as well as safe. So it's really the foundation of where therapies come from. And that moves us into translational research, where we can then start testing some of these different therapies and improving upon them. So, you know, we have excellent cores and researchers here at Vanderbilt that can not only take existing drugs, but modify them, right? And maybe improve their lifespan in the tissue or improve their ability to act on their receptor. And then we also have amazing engineers on campus that can help us perhaps package them in such a way for sustained release. You know, we're talking about eye diseases here. So oftentimes we're having to deliver this therapy into the eye. And we haven't discussed it, but in AMD, some of these mechanistic studies helped us to realize that the major molecule causing wet AMD, which is neovascular age-related macular degeneration, where you have new blood vessels forming and they're leaky, and that causes blindness. The major molecule was called vascular endothelial growth factor, or VEGF. And by identifying that, we realized as a field that if we could limit the amount of that, because it's increased in the disease, that maybe that would prevent some of this pathology. And in fact, it does. So patients now can go in and get intraocular injections of anti-VEGF, and it blocks VEGF, and the vision is retained. So that model is something we use frequently in translational research. And, you know, testing different potential therapies, and sustained release allows us to inject in the eye less frequently, which is really important. Nobody likes to have anything put on the eye, much less into the eye. So, you know, I'm working with colleagues. We have for example, a patent on my molecule, patent on some particles, and we've put them together, and we've been able to patent that. Or in the process of doing so, with the hope of moving that to the clinic. True bench to bedside research is something we want to do here at Vanderbilt. And that's the pathway, right? So identifying the mechanisms, testing rational therapies, optimizing them for the clinic, showing safety and efficacy so that we can move hopefully to clinical trials and really help patients. That's our ultimate goal, and that's translational research, right? Translating from bench to bedside.
And I think that's so important to understand, is just what is that whole continuum and how important is each part of that, especially the foundational mechanisms, understanding that for which you can target. Dr. Furman, what are the frontiers in AMD that you're really excited about that the field is uncovering right now?
Yeah, I mean, one really important research direction has evolved from developmental biology, which is stem cell research. This is a very important approach, and one course of hopefully potential treatment in the future. So developmental biology helped us to at least identify some of the mechanism how we can differentiate or promote the production of RPE cells from more immature cells that could even come from your own body. And so this is basically done in a dish. And there are clinical trials now underway where these RPE-derived stem cells are implanted into animal models. They started out as animal models, but they are also used in a small number of patients. And so efficacy and safety experiments are underway for that. And there are challenges with these approaches. They're very promising, but there are also a lot of challenges. For example, if you inject a suspension of RPE-derived, stem cell-derived RPE into the eye, they can have a hard time integrating into the tissue that is already there or into the eye. Another really nice approach is to seed these cells and grow them on a scaffold and to implant that. But that also needs specific surgical manipulations, which can be a little bit risky. So there are also more advanced approaches, and I really want to warn also about maybe companies that might offer bone marrow-derived stem cell treatments. This is not so well certified, I would say. So I think this is being offered, and sometimes I get asked about that. So this is something where you really want to educate yourself. So I'm very excited about the stem cell derived research directions. I don't work directly on them. What we are trying to do is we are trying to promote or awaken a stem cell potential of the endogenous RPE in the adult eye upon injuries, so that the RPE can basically repair itself or even try to regenerate itself.
Can I just add to that?
Definitely.
So Dr. Calkins and I each separately and somewhat collaboratively are working on some of the cell transplantation work that Dr. Furman just referenced, interested in induced pluripotent stem cells. So that's taking fibroblasts, skin cells from patients and converting them into stem cells, making them able to convert into any cell type we want. And then we push them towards the retinal ganglion cell fate. Those are the cells who extend axons through the optic nerve to the brain. So those are the cells that are affected in glaucoma and in traumatic optic neuropathy and other optic neuropathies. I do agree with Dr. Furman. I think it's a really exciting area of research. The clinical trials that are ongoing right now that are working with cells, those cells typically are staying just within the eye and they're releasing factors. So it's really more of a neuroprotective approach at this point. And you could almost just treat with neuroprotectors, but they're not truly integrating most of the time, not in the trials that are currently ongoing. But that is an area of heavy investigation for the photoreceptors as well as for the retinal ganglion cells, trying to successfully foster survival and integration of these neurons into degenerative retina to restore sight. But we are many years off before that's going to reach a patient. But it's a very exciting area of research right now, and I have a lot of hope and faith that we will get to patients someday down the road, but we're not there yet.
No, it sounds like some very exciting potentials for patients. And Dr. Shai, I want to get back to you on the frontiers in cataracts, and maybe you can even comment further on just the time period from discovering a mechanism to potentially getting into the clinic and what the length is, just so people understand. Your sound is not coming through for some reason. We can't hear you for some reason, Dr. Shai.
Well, while he
he
gets his sound
yes.
going, I can speak to the amount of time that it
Yes,
takes.
thank you.
Yeah. Oh, there you are. You're back. Never mind. Take it away.
A phone call came in and it uses the same audio system as my computer. So sorry about that. Oh yeah, so the time frame. Erin, that's an excellent question. And I've been working on cataract for most of my career. And my family, some of who are listening in, have asked me, you haven't solved the problem yet? And so the time frame can be very, very long. It's very, disease dependent. So sometimes biochemical mechanisms just sort of fall out and they're easy to manipulate, relatively speaking. And so, you can come up with a therapy or envision a therapy rather quickly. On the other hand, some of these age-related processes are challenging to understand the mechanisms behind them. And even more challenging is how do you reverse, right? Or how do you reverse a process that has taken decades to occur? And in my opinion, the answer is you're probably not going to reverse it, but you can delay it or prevent it. And so, from my point of view, the areas in cataract research are, first of all, prevention or delay by either diet or some antioxidant type therapies. The other area of active investigation in cataract research is after the cataract surgery occurs, there's what we call a secondary cataract that can form called posterior subcapsular opacification, or PCO. And this requires a laser treatment after the cataract surgery, anywhere from 2 to 5 years after the surgery. So an active area of research is how do we prevent this PCO from happening? How do we, and of course, how do we treat it effectively so you don't have to go through multiple laser treatments after cataract surgery?
Thank you so much for that. And I think that's so important to understand just how complicated biological mechanisms are and how long and how large the scientific field is that's all coming at these problems to solve them. And hopefully that speed is increasing with technology. Dr. Rex, one more question for you before we get to some of the Q&A. Are there disparities in eye disease that we should be aware of?
Right. Certainly in glaucoma, my area, one of my areas of research, it's prevalent in the African American community. And it's a big challenge to diagnose it early. And unfortunately, we don't have the representation of African Americans as ophthalmologists that I think that we need. So, we're all more comfortable. I think the data shows that we're more comfortable talking to physicians who look like us. And people want to stay in their communities. So I think there's a great need in ophthalmology to improve our diversity on the physician level and improve our presence in the communities where glaucoma is most prevalent. So there's a real need there for us to reach out and to build relationships and to increase our diversity there, so that we can reach out to that patient population more effectively. And then within the clinics, we need to do a better job of educating in the communities too, to let individuals know glaucoma is sneaky. You know, because it's peripheral first, we don't tend to use our peripheral vision a lot. You can lose a lot of it before you realize it. So really important to educate individuals in the community that if they have any family history at all of glaucoma, they really should be checked by an ophthalmologist, specifically an ophthalmologist. And, you know, just to get their IOP monitored, to make sure that it's caught early, because if it's caught early, we can treat early and the prognosis is better. So there's a lot of need there for education and outreach in the community.
Oh, that's so important, and just that early detection as it continues to get better and better, connected to that issue. So we're moving to some of the audience Q&A. So if you have questions in the audience, please place them in the Q&A at the bottom of your screen. The first question I want to start with, we were actually talking about this before the event started. What is the current state of research into the decreasing prevalence of myopia? Dr. Rex, would you want to start with that, or Dr. Furman or Dr. Shah? I'm not sure who the best would be to answer that.
Well, I can say a little bit, maybe we can bet it to Dr. Shah, because I know he mentioned he's doing some work in this area, but you know, gratefully, we have some really great seminars that we host here at Vanderbilt Eye Institute. We've been able to bring some individuals in and go to conferences. So even though I don't work in this field directly, I do keep up with it. And it's shocking, frankly, the exponential increase in myopia cases worldwide, primarily in Asia. And there's some controversy in the field as to why that is happening and what controls myopia, and we're learning a lot more, but certainly the data that I've seen, the research that I've seen, does indicate that our screen time is contributing, and particularly children, as they're developing, we need to make sure that they're outside, they're exposed to the full light spectrum, they're seeing with depth perception, and, you know, items that are close and far, you know, seeing their friend amongst a forest of trees, for example, and really utilizing the visual system, probably a couple hours a day would be ideal, is what I'm learning. But again, I'll bat this now to Dr. Shai.
Yeah, I think I don't have a whole lot more to add other than to say that we're currently working with a group at the University of Alabama, Birmingham, who are world leaders in understanding eye development and the formation of myopia. It's very, myopia is very dependent on the shape of the eye and how the eye develops the proper distance from the front to the back. And interestingly, the exact signals that occur during development to say, okay, you've grown enough, you're at the exact proper distance and size, those signals are not entirely understood. So that's what we're trying to understand, to learn more about myopia and why the eye doesn't focus properly.
Dr. Furman, we have one question about, are there new treatments to stop the progression of wet AMD?
I think the general approach is the same, to try to inhibit the excessive vascular developing, but there are new treatments where you don't need to get monthly injections, which also can cause complications. So these new treatments can be done, injections can be done every 3 to 4 months, if I'm correctly. So yeah, I think it's important to ask your doctor also what is available and what they do.
Thank you. And we had a question about how is eye pressure connected to eye disease? Is it connected to just one? Is it connected to most? Dr. Rex?
Right, so eye pressure is typically associated with glaucoma. And it's thought, although it's the active area of research, but there are proteins in these cells of the retina that are on the cell surface, and they open up and allow ions to travel, and that can cause signaling changes. And some of these, they're called receptors, are in their channels, are mechanosensitive. So increased pressure can cause activation of these, and that can cause signaling changes in the cells, and that can initiate some processes that are detrimental. In addition, that pressure is thought to particularly affect where the retinal ganglion cell axons exit the eye and start forming the optic nerve. And that specialized area that that optic nerve had is thought to, when there's pressure, kind of impinge upon the axons, cause physical detriment to them. And it's also a highly metabolically active area for a number of reasons, and I won't get into the details today, but it's thought to really affect the function of the mitochondria and metabolic activity, and really stressing those axons metabolically as well. So so the physical push of elevated pressure causes molecular changes that stresses those retinal ganglion cells, resulting in their degeneration.
No, that makes so much sense, just how that can be a factor in a long period of time.
Yeah, and then, sorry.
No, go ahead.
Pressure can, there's also a faster transient of pressure that occurs in trauma, particularly from blast exposure, thinking in the context of the military, whether it's IEDs or the breacher environment, but exposure to explosives and those blast waves, that pressure differential can cause shearing damage of those long axons. And so in addition to the long-term effect of slightly elevated pressure, and even higher pressure in short time can cause a different type of damage that can also be permanent to those neurons. Sorry, done. Thanks.
No, that's important work that you're doing with the military on that. And Dr. Shai, there's a few questions in the Q&A about cataract surgery. I'm not sure if you have, as not a clinician, if you can comment on that, but one is just when in the formation of cataracts do you have surgery? And then what is the recovery like from that surgery, if you can answer those?
Yeah, so again, I'm not a clinician, so your ophthalmologist can answer these questions with much more accuracy than me. But my understanding is that the recovery from the surgery is very rapid, just a few days. I know there is an extended treatment of antibiotic eye drops, but the recovery of your vision is very fast. When in the course of cataract formation is surgery appropriate? That's a discussion to have with the ophthalmologist. I know, when the last time I was at the ophthalmologist, that I had the beginning of cataract form, but I don't notice any change in my vision. So, I'll put off the surgery until I notice some effect on my vision, and that's typically, I think, when the ophthalmologist would recommend surgery. There was another question related to this about monovision and the after cataract surgery. Is there a way to avoid having to wear glasses? And again, I'm not an ophthalmologist, but my understanding is that it's difficult to correct your vision perfectly after cataract surgery so that you can see both near and far. So oftentimes the case is that you will still need reading glasses after cataract surgery. There are newer technologies, newer types of intraocular lenses, those artificial lenses that they put that can actually focus a little bit near and far, but they're still not perfect. And so chances are you will probably need some form of reading glasses even after cataract surgery.
Thank you so much. That's very helpful. And We have an interesting question about there's vision changes in Alzheimer's disease and potentially other neurodegenerative diseases. Is research being done in this area?
I guess I'll take this. Yes, absolutely. It's been an area of interest for a very long time. There are a number of groups investigating these intersections. As I mentioned earlier, the retina is brain. It's a piece of the brain that's accessible to us to be able to see. Just as we see out, we can look in. And it's so just as there's neurodegenerations that affect the brain, they're going to affect the retina in similar ways, and we can learn from each other. There's a lot of commonalities there and a lot of crosstalk. So yes, very, very hot area of research.
Yeah, just again showcasing the body as an entire system that's affected broadly by different impacts. And one question was, is there hope for future treatments for individuals who are blind from cone death and photoreceptor degeneration?
Right. And before I answer that, I want to say one advantage, though, of the retina being so accessible and that there being changes commonly is that we can potentially use biomarkers of, changes in the retina or the lens as biomarkers for neurodegenerative changes like Alzheimer's. So there's potential there, which is exciting. But yeah, in terms of cone degenerations and photoreceptor degenerations in general, some years ago we had great success that continues in terms of gene therapy to treat inherited retinal degenerations, and those typically affect photoreceptors, mostly the rod photoreceptors. But to the individual's question, some of these are cone dominant retinal degenerations. And so we do have precedent for success of delivery of the corrective gene, so the wild type form of that gene into the appropriate cells to restore sight in individuals who have an inherited retinal degeneration. Now, in that gene therapy, the cells, this happened to be a mutation in a gene that expresses a protein specifically within the retinal pigment epithelium. So not within the photoreceptors, but it's an enzyme that's really critical for photoreceptor function. Since then, there have been a number of clinical trials initiated for photoreceptor directed gene therapy, again, replacing or adding in wild type form of the gene. So this is going to work in the case of a recessive mutations where you have loss of function, and so we can restore it by adding in the functional form of the gene into those cells. So it's an active area investigation. It's a lot harder to deliver specifically with high enough copy number to show therapeutic efficacy into the photoreceptors, even more so in the cone photoreceptors, although some promise is the fact that the vectors that we use to deliver the corrective gene in people, it's easier to transduce the macula, which is the cone dominant region of our retina that we use the most. So that gives greater promise for cone delivery of these genes. So there's a number of clinical trials ongoing, a lot of active research, a lot of companies interested, which helps a lot. So a lot of promise coming down the pike.
That's exciting. And that really leads into our last question to close out the discussion. Where do you all see prevention treatment heading in the next 5 to 10 years with the exciting research that's going on? Dr. Shai, do you want to start, and we'll go around and end the event?
Yeah, I think, what I heard as a theme coming up over and over again in our talk today was early detection. And if we can detect any of these diseases we're talking about early enough, then we can start trying to prevent or delay serious visual outcomes. And so treatment, you asked about treatment specifically, I think, changes in diet. If we can have healthier diets, rich in antioxidants, this will help all parts of our body, but will certainly help the visual system. And then as Tonya just mentioned, Gene therapy is coming. I heard a presentation at a recent conference from one of the leaders in the field, and there's so many active areas of research in gene therapy, different ways to deliver and target. It's super exciting, and it won't be tomorrow, it won't be next year, but it's coming.
Thank you so much, Dr. Furman.
I think what I'm really excited about these technologies like RNA sequencing, for example, that help us to dissect out the importance of each tissue that is involved. So, for example, when we look at AMD, it's not just the RPE itself, it's also the environment, the retina and the choroid, for example, the extracellular matrix. And so if we can kind of figure out, it's not just the changes that are happening in the RPE, but also in the surrounding tissues and the pathways that are involved. So it's a very complex problem that will help a lot too. And I think this is where more information will be very important. Also within the RPE itself, there are regional changes that make certain cells more susceptible to damage, for example, the macular region compared to the more periphery of the RPE. I think this will be also very important for treatment to kind of have a more complex vision of this.
Thank you. And Dr. Rex.
Right. So we study complex diseases. And I think what's really exciting is that there's been this development from multiple research areas, multiple areas of expertise, so that we're all kind of converging together right now with advanced imaging, AI techniques, and machine learning, advanced molecular techniques. And all of this together means that we can really start addressing these complex diseases in ways that we never could before. So I completely agree with Dr. Shai. We have to identify them earlier, and we have the tools available now to be able to do so. We're really, pushing the edge of our sensitivity for biomarkers to be able to do that. It's much easier to treat than it is to restore something that's lost. Mm-hmm. So getting in there early, we have some great approaches now for sustained delivery of some really exciting neuroprotective agents. So being able to treat, I think, is coming to the forefront. And despite all of that and recognizing that that's the best approach, unfortunately, there are patients who have lost vision. So it's really important to keep pushing the field and restoration and regeneration. And we are making strides there. So, it's going to be a staged thing, but right now it's just, we have all the tools available. It's not like decades before where we were sitting there in frustration, and now we don't have to do that. It's the field's moving forward at a rapid pace these days. So it's very exciting.
Thank you so much, Dr. Rex. And thank you so much to all the panelists today for really providing their insights into the exciting time in eye research and what the future holds for treatment and prevention. And thank you to the audience for attending. We will be posting this recording on YouTube and sharing the link with everyone. And we hope you attend another Lab to Table conversation in the future. So thank you all very much, and we hope everyone has a wonderful day. Bye, everybody.
Thank you.